Our speaker today is Dr. Henriette van Praag of the Stiles-Nicholson Brain Institute at the Charles E. Schmidt College of Medicine at Florida Atlantic University. She will share her insights into the relationship between exercise and memory function. For some technical reasons, the recording of the first few moments of her presentation were lost, so we jump right in following her opening remarks.
Has different sections that are responsible for different functions, such as motor behaviors, sensory sensation, seeing, memory, navigation, planning, and judging. And here in the next slide, you can see in a little bit more detail the functions, the big variety of functions of the different brain areas. So you can see, for example, the occipital lobe in the back is important for vision, the brainstem for breathing and consciousness, the temporal lobe, which contains the hippocampus, our brain area of interest, is important for memory, but also for hearing and understanding language. And then the frontal lobe is important for judgment, speaking, movement. So a very complex and important organ that consists of cells, of nerve cells that are responsible for executing all these complex functions. And you've probably already learned in your biology class that a neuron consists of a cell body which contains a nucleus, dendrites, which are important for receiving information from other neurons, and that the neuron transmits its messages electrically and then chemically by release of a neurotransmitter from the axon terminals to other nerve cells. So neurons are essential for our cognitive, for thinking, for emotion, for mood, for motor and sensory functions. So without them, our brain cannot think, feel, control the body. And unfortunately, with certain degenerative diseases or during aging, neurons can atrophy or die, and the brain function will decline. And this is particularly important for our brain area of interest, which is the hippocampus. The hippocampus is essential for memory function. It is also among one of the first brain areas that is affected by normal aging and also by Alzheimer's disease. You can see this is a section through the human brain here on the right-hand side, and you can see where the red circle is, that is that little small area there, that is the hippocampus. And without it, it is not possible to form new memories. So that is sort of the not so good news, that with aging, that brain area can atrophy and decline, and it can lose neurons, and it can lose function. However, it is also a unique brain area because it can give rise to new neurons in the adult brain. This idea that the adult brain can give rise to new neurons, however, was not always well accepted. It was actually very controversial for many, many years. The dogma was that no new neurons could ever be formed in the adult brain. The famous neuroanatomist Santiago Ramon y Cajal stated that once development was ended, the fonts of growth and regenerations of the axons and dendrites dried up irrevocably. In adult centers, the nerve paths are something fixed and immutable. Everything may die and nothing can be regenerated. But he also added to that it was up to future generations, future researchers, to try to reverse that harsh decree. And indeed, we now know that there are two small areas in the adult mammalian brain that can give rise to new neurons. One is an area called the subventricular zone. So the brain contains these pockets of fluid. They're called ventricles, and the lining of the ventricles contains specialized cells that can give rise to new neurons in the olfactory bulb, in the nose, the brain area is responsible for smell. So it's not in the nose, but it's the brain area that is responsible for smell. And then we can see here where the second area just came up, that is the dentate gyrus of the hippocampus, a subfield of the brain area that's important for learning and memory, that also can give rise to new neurons. And that brain area, as I mentioned, is our focus of interest. Now, I also told you that idea that the adult mammalian brain can produce new neurons for many years was very controversial. Why was it controversial? It was because Joseph Altman, who made the original discovery with his colleague Shirley Das in the early 1960s, did not have the modern tools that we have today. So what he used was something called tritiated thymidine, a radio label. And you can see these black dots here in the what we call the granule cell layer that are labeled with radioactive material. And those, he said, were new neurons. Why did he say they were new neurons? Because the label that he used could only be taken up by dividing cells. That was one reason. So cells that were supposedly newly generated. And the other reason he said was that this brain area does not have many so-called glial cells or support cells. So that's why these cells could only be neurons. But his work was largely ignored for about 30 yein until two things happened in the mid-1990s. First of all, it was found that in birds, in the canaries, it was found that when they learn a new song, they grow new neurons in the brain area that produces song learning. And in another bird that you can see here, this is called a black-capped chickadee. These birds, when they start to store seeds for winter, they grow a brain area, they grow neurons in a brain area that's very similar to the hippocampus. And, oops, sorry. So when these birds are storing seeds, they have more new neurons that are being grown in the what we call the avian hippocampus, suggesting that there is a relevance for memory function with regard to the production of these neurons. So that sparked the interest of researchers that work on the mammalian brain. In addition, other researchers found around the same time that the adult brain contains so-called stem cells, undifferentiated cells that could be taken in a dish, and then they could become actually new neurons. So, and then a third thing happened in the mid-1990s is that there were now new tools by which you could actually more confidently identify new neurons in the adult brain. So here now we have a specific marker for neurons in green. We have the cells labeled with, not with this radioactive tracer, but with something called BRDU that labels every dividing cell in the body and in the brain. And then you can now see that here are these orange tinged cells in the embedded in between the green cells that can be considered new neurons. However, even with this new technique, and also with more powerful microscopes that really allowed for quantification. Now here on the top panel, you see the BRDU labeled cells just as brown dots, so the cells could be visualized and counted. They could be more convincingly identified as neurons. But sorry. Moving. But it was still not clear, or there was still some doubt in the scientific community whether these cells were actual neurons. Did they really have dendrites and an axon? Could they fire action potentials and release neurotransmitters? Were they really similar to what I showed you at the beginning of the lecture, how a neuron looks? Because what I just showed you were just these nuclei, these dots. So to more convincingly show that these cells are new neurons, we used yet another method called a retrovirus, which was directly injected into the brain of mice to label dividing cells. And with this method, we could label the entire cell, not just the cell body. And now we could show how these cells grow over time, how they're small on day 2 after labeling, and finally become, after about a month, these really beautiful new neurons in the adult mouse hippocampus that you see here in green. So now you can not only see just these little dots, these little nuclei, but you can actually see the cell bodies, the dendrites where they receive information, and also all the axonal processes. So, as I mentioned, adult neurogenesis doesn't occur in every part of the brain, only in the subventricular to olfactory bulb area, and in the subgranular zone of the dentate gyrus of the hippocampus. That's one thing that's special about it. The other thing that's very special about adult neurogenesis is that it can be increased and decreased by a variety of factors, including our own behavior. So with exercise, as I'll show you, there is a profound increase in the number of new neurons. It can double or triple. Whereas with things such as aging, stress, high-fat diet, so excessive calorie intake, excessive alcohol intake, there is a reduction in the number of these newly born neurons. So how was this discovery made? How was this found? So a researcher called Gerd Kemperman in 1997 showed that if you house animals in a so-called enriched environment, which is, as you can see here, a bigger cage with lots of toys, more friends to play with, an activity wheel, more opportunity for social interaction, more opportunity for learning, then these mice have more new neurons than mice that are housed in a standard cage in their hippocampi. And then we try to figure out what is the component of this enriched environment that increases the number of these newly born neurons. And what we found at the time was indeed that if you only give the animals a running wheel, that is sufficient to produce more new neurons. So to show you that a little bit more clearly, we did another experiment where we had animals in control conditions, running conditions, what we call enrichment only, or toys plus running. And again, we gave them injections of BRDU to see the number of new neurons. So here you can see the different conditions. That on the top shelf, there's the empty cage, then the cage with the wheels, the cage with the toys, and the toys plus the wheels. And what you can see, looking at the green dots, which are the new neurons or the new cells, that only if there's exercise, only if there's running, do we get about a doubling of the number of these newly born neurons. So we have shown quite convincingly that exercise enhances what we call adult hippocampal neurogenesis, which is a big word for the production of new neurons in the adult brain. So, and we also show that this increase in these newly born neurons is associated with improvements in learning and memory. How did we do that? Now we, again, housed animals either in what we call a standard cage or in a cage with a running wheel. And then we tested them in a mouse memory task called the water maze. In this maze, the mice have to swim to find a platform that's hidden a little bit under the surface of a pool that's made murky with a little bit of non-toxic paint. After a few days of training, they train for about 4 or 5 minutes each day. They get 4 trials of about a minute. And if the mouse doesn't find the platform, we either guide it to it or and then we take them out of the pool. The mice will learn how to find this. And what you can see that if you house animals with a running wheel for about a month, and it doesn't matter if they're young, if they're middle-aged, or if they're aged mice, the runners will do much better on this test. As you can see, the little trace of the running mouse finding the platform much quicker than the control mouse that circles around the pool for a much longer period of time and has trouble remembering where that platform was located. So based on this, we concluded that mice, at least in mice, exercise improves learning and memory. And studies by people in humans have shown similar findings. Now, another memory test that we do is what we call pattern separation. I won't go exactly into all the details of this, of the reasons why we do this test, but what you can think about is that the number of neurons that come from the cortex are fewer than the ones that we have in the dentate gyrus, where new neurons are being born. And that allows for more finer storage of information, which allows us to be able to distinguish between events or stimuli that are very, very similar. For example, if you go to the mall and you have to find your car in the parking garage, it's very difficult because a lot of cars have similar colors and similar makes and models. So you have to be very good at distinguishing highly similar, what we call stimuli, to find your car. And this is a very specialized form of memory that is mediated by the dentate gyrus of the hippocampus. And it's also a form of memory that declines very soon with aging. So we train mice to literally touch a computer screen with icons on it that are very similar to learn this test. I'll show that to you in a little movie. So here the mouse is in a magazine. He gets a reward every time he touches the screen. He gets a little drink of condensed milk. And once he's learned this test, then we can challenge him to make it easier or more difficult. And then we can ask the question, is increased neurogenesis associated with improved pattern separation? So here we have challenged the animals by having them have to choose or find a reward that's associated with one of the icons on the screen when the icons are either far apart or close together. And when the icons are far apart, the task is easy. When the two icons on the screen are close to each other, then the task is very difficult, with only one of those icons being rewarded. And what we found is that when the task is very difficult, when the icons are close together, to do well on this test, the animals that are housed with a running wheel do better than the animals that are housed under what we call sedentary or control conditions. So less trials to criterion, what we call, is better. And the number of trials showed a trend towards an association with the number of new neurons that were being produced. So this is another type of memory task that that shows the importance of physical activity for brain function. So what I've shown you so far is that running increases the number of newly born neurons, and it also improves learning type and memory, different types of learning and memory. Now, one of the follow-up questions that we asked, and again, I won't go in great detail about this, but I just want to show you a few of our pictures and data points about this, is, well, what if we had a magic pill? What if we could just boost the number of these neurons in the dentate gyrus of the hippocampus? Would that be enough to improve learning and memory? Or do we actually need something else? Is there like a network that these neurons are a part of? And if we exercise, do we not only increase the number of newly born neurons, but do we change more things in the brain? Do we change the brain's network? So to answer this question, we used another method where we put a special, now even a more special virus into these newly born neurons and combined it with another virus that we used as a tracer to look at the circuitry. And we found that these newly born neurons are connected to many brain areas that are important for learning and memory. And this method that we used to trace their connections allowed us to visualize all the different areas that provide input or connections to these newly born neurons and how those develop over time. So, for example, we found that initially the connections of the new neurons are local, they're within the hippocampus. But when these new neurons grow up in the adult brain, when they're about a month old, they get input from an important brain area that I've already mentioned, the entorhinal cortex, that I showed you was linked to the pattern separation memory function. So what happens when the mice exercise? How does that change the inputs to these newly born neurons? So again, we do the treatment with these two viral vectors. And what you will see is that under control conditions, you can see the red cells that are the ones that project to the newly born neurons from what we call the LEC and the MEC, the lateral and medial entorhinal cortex, are rather sparse in comparison to the exercising mouse, where there are many, many neurons here in red labeled in red, that project to the adult-born neurons in what we call the dentate gyrus. So these red cells are not new neurons that are being generated. Rather, they are cortical neurons that are labeled, that are connected. So you can understand that exercise doesn't only increase the number of these newly born neurons, it also strengthens the pathways by which they are connected to other brain areas. So with that said, running increases adult neurogenesis locally in the hippocampus, but it also increases what we call the plasticity of the network by which they're connected throughout the brain. And of course, as I already mentioned a few times, it enhances learning and memory. Now, we still, though, are trying to understand why exercise improves what we call cognitive function. I talked with you already about the production of new neurons, but we know that other things happen with exercise in the brain too. There is more blood flow in the brain. There's an upregulation of what we call neurotrophins or growth factors in the hippocampus. There are changes, neurochemical changes, and there are changes in the morphology or in the structure of existing neurons. So a lot of things happen actually in the brain with the exercise beyond the production of new neurons. And why is exercise so powerful? So one of the reasons we think is because there is a link between other organs, in particularly muscle and the brain. And how do we know that? How did we get to that idea? So there are a couple of reasons. I will just give you a little overview of some of the concepts that led to the work that now is showing that there is a link between peripheral organs and the brain that may mediate some of the effects of exercise on the brain. One is a very complicated word called heterochronic parabiosis, but basically what it means is that the bloodstream of an old mouse was linked to that of one mouse was linked to another mouse. So in this case, it was the blood stream of an aged mouse was linked to that of a young mouse, and it was shown that that reduced the number of new neurons that was produced in the brain of these young mice, suggesting that there's something in the blood of the animals that could do that. Also, if researchers took blood from running mice and infused it into mice that were not active, they recently showed that improves neurogenesis and memory, again showing that there's something in the circulation. And then there's other evidence that if you activate with pharmacology, with drugs, the skeletal muscle of mice, that could mimic the effects of exercise. So here you can just see in this little cartoon how we're thinking about this. So you have the animals run and you can collect their blood, and then in that blood there is something that can affect the function of the brain in a positive way. But of course, something has to be released into the bloodstream. So the question is now, which organs and which factors are involved? So in recent years, a lot of factors have been identified from different organs, from the muscle, from the liver, and also from what we call adipose or fat tissue. And how did we get involved in this study between communication between the periphery and the brain? So we studied this because we got interested in compounds that drugs that could activate skeletal muscle and that seemed to mimic the effects of exercise. Specifically, there was a compound called AICAR that was injected in mice, in sedentary mice that were housed just under controlled conditions. And then when the researchers put those animals on treadmills, they had a greater endurance than sedentary animals. I have to warn you that AICAR is now a banned substance. It's on the doping list. It's not allowed for athletes. And also, it is actually quite dangerous. Even in mice, we can see that with prolonged intake, it causes inflammation in the brain. However, with short-term use, it actually can, in mice, it can mimic the what we call the central effects of exercise, the effects of exercise on the brain. It can increase neurogenesis and cognitive function. And we think that that happens by activation of muscle. So we see improved performance on what we call the water maze. The animals get the drug, find the platform more quickly and remember it better. And there's an increase in the number of these newly born neurons. So in addition, if we knock out a special cell, a special receptor for AMP kinase in muscles, then the effect of this drug on memory is no longer there. The drug no longer is effective. So this led us to think that the benefits of exercise for learning and memory might originate from the muscle. So how did we try to study that in more detail? So what you can see here is a little dish with muscle cells in it. These little fibers in it are muscles. We call them myoblast cells. And we treated them with the same compound, with AICAR. And then we took what we call the conditioned media, or the liquid that these cells were growing on. And we took that and we put that on neural stem cells, so cells that were derived from the brain. So we treated the cells, either with what we call vehicle, control substance, or with AICAR, and collected the media. And as I mentioned to you, you can take these adult neural stem cells and grow them in a dish. So here at the top, these blue dots are brain cells that are what we call undifferentiated. And if you give them certain compounds, they can become neurons, oligodendrocytes, a type of glia, or astrocytes, another type of glia. So what we saw when we placed the media that we took off the muscle cells that were treated with this with AICAR and put it on the neural cells, we saw that there were more neurons in the dish than in the vehicle condition, suggesting that there is something in that soup that comes off the muscle cells that affects brain cells, making a link between the muscle and the brain. So what's in that soup? So that's something we looked at in the lab. And again, I won't go into all the details of this, but we identified something that we call a myokine, a factor that is released from muscle. It's a lysosomal enzyme that is called Cathepsin B. So you can see that if you that with running, there is an increase in Cathepsin B in mouse muscle and in plasma. So you can see that there's increase in gene expression, and there's increase in the protein levels of Cathepsin B, and there's an increase in the blood. Then when we looked at mice that do not have Cathepsin B in their bodies, they are so-called knockout mice for Cathepsin B, for this myokine, and we tested them on a bunch of behavior tasks, one of them being the Morris water maze, which I already told you about, which is the mouse memory test. But we also tested them on their motor behavior, on measures of anxiety, on measures of depression. And what we found was that the mice, the knockout, what we call the Cathepsin B knockout mice, no longer did well on the Morris water maze, on the mouse memory test. In addition, the knockout mice did not produce more new neurons when they were housed with a running wheel, suggesting that this factor, Cathepsin B, is important for adult neurogenesis, for the production of new neurons, especially under running conditions. So then the next question we asked was, well, if we see this first in the dish, treated with the compound AICAR, that there's a release of this factor, then we see it in the mice, and it appears to be important for learning and memory. Does this have any significance for humans? Does exercise increase Cathepsin B levels in humans? So here we had two groups. Well, these were college students that were all couch potatoes. They didn't exercise at all. And they were assigned to either a control condition or a treadmill condition. And in the control condition, they were walking twice a week at 50% heart rate for about 10 to 15 minutes. Whereas in the treadmill condition, they were jogging or running 3 times a week at 70 to 90% heart rate for about an hour. And this intervention lasted for about 4 months. And what was found after the intervention, that people, or those people that had a good increase in their Cathepsin B plasma levels in blood, also were more fit. And in addition, those people that did well on or had a good increase in their Cathepsin B levels over time also did better on a memory task called the Ray-Ostrich complex figure task. And you can see this sort of funny looking figure here on the next to the graph. And what happens is they first give people a piece of paper to copy this figure onto, and then an hour later they have to redraw the figure from memory. And those people who did better on that test also had an elevated Cathepsin B level, suggesting that this myokine is relevant to learning and memory, and maybe also important for older adults. So that was another study that we did is where, in collaboration this time with Ozioma Okonkwo at University of Madison, Wisconsin, who has a cohort of elderly individuals, some of whom are at increased risk for Alzheimer's disease, which you probably are all aware of is one of the main forms of dementia that happens with old age. And these people were either at increased risk because they had a direct relative that had the disease, or they had a gene called APOE4. So the average age here was 65, males and females, and this time the exercise intervention was 6 months. So here you can see the design of this study. People come for a baseline visit with the researchers. They get a blood draw, they get some other tests. They also have cognitive testing, and then they're assigned to either usual physical activity, they get a booklet, or they have supervised enhanced physical activity for 150 minutes a week for 26 weeks, and then they come back for another blood draw and more cognitive testing. So here you can just see they get the controls are walking, it's the woman walking the dog, and then the person on the treadmill exercises with increasing intensity over time. And as I mentioned, you have the blood draw at baseline, and then you have the blood draw again 6 months later. And we were sent the blood samples, and what we saw was that those people that, the elderly people that had the enhanced exercise intervention had higher levels of Cathepsin B in their blood compared to the control group. In addition, those people that had a good increase in their Cathepsin B levels also did better on a on another what we call hippocampus dependent memory test. This is the California verbal learning test, where people have to remember a list of words. So those people that did better on the task had a better increase in their Cathepsin B levels in these older adults. So altogether, what I've shown you is that there's an important link between the periphery, so they're peripheral factors, and I've just gave you the example of one called Cathepsin B that play a role in learning and memory and may help us understand how exercise influences brain function. Now, Cathepsin B is not the only one. You may have heard of irisin or lactate. Those are other factors. And I also mentioned that there are factors that are released from the liver and from fat tissue. So there's likely not one factor that does everything, but more likely all these factors interact to affect brain function. So in conclusion, what I've shown you today is that running increases neurogenesis and memory function. Exercise also changes the network of these newly born neurons. I also just showed you some of our research pertaining to the link between the periphery and the brain, and the idea or the concept that some of the benefits of exercise for cognitive function could originate from peripheral organs. I showed you also that what we call CTSB or Cathepsin B is what we call a myokine, which is a factor that's released from muscle, and that it's associated with memory function, not only in mice, but also in humans. And of course, there are many other factors in the periphery and in the brain that are released as a result of exercise, or that mediate the effects of exercise on the brain, that remain to be discovered. And again, these are some of the factors from the different organs, from muscle, adipose, and liver, that can affect neurogenesis, cognition, mood, synaptic plasticity, and some of these neurotrophins that I briefly mentioned. And with that, I'll be very happy to take any questions you might have.
Thank you very much.